Good morning students. In the last class we have discussed about breeding of Anthurium, and in today's class we will discuss about breeding of Gerbera. And as you all know the importance of Gerbera, so the Gerbera flowers is one of the most prominent and highly sold flowers in the international market, and it is prominent, that is it plays about stands in a 5th place in the international trade. And the Indian flowers of these Gerberas are generally exported to European countries, and also these flowers are very attractive, and also the flowers can be easily rehydrated, that is after harvest, and also for keeping for many days. So immediately we can place it in a water and it can rejuvenate again, and it can rehydrate so water, and it can last for a longer time, and the freshness can be maintained. Then apart from this, the flowers are generally of different colors, and also so wide range of colors are available in case of Gerbera due to its wider genetic base, that is about 45 different species of Gerberas are present. And apart from that, if any colors which is absent in case of Gerbera, so we can produce those colors by the process called tinting. So this is Gerbera is one of the best flower for tinting also. By tinting we can get a desired flower colors in case of Gerbera, and can be used in a decorations and other things. So mainly this Gerbera flowers are generally used for cut flower purposes, and also for floral arrangements and other things. So generally these flowers are used. And apart from this, generally this cultivation of these Gerberas are generally cultivated under protected cultivations like polyhouse conditions, and also in the shade house condition we can go for cultivation of Gerbera, provided so there is no water stagnation in the shade nets, so we can go for cultivation. Then apart from that, some of the varieties of Gerberas can also be cultivated in the open field also, like the IAHR released a variety called Karka Krishika. So it's a yellow color variety in case of Gerbera, so it is suitable for open field cultivation, so we can directly cultivate in a open field. So this is some of the brief about Gerbera. So coming to the breeding aspects of Gerbera, so as you all know the botanical name of Gerbera, so that is nothing but Gerbera jamisoni. And commonly these Gerbera flowers are called as Transvaal daisy, Barberton daisy, and also African daisy. This is based upon that is Transvaal daisy because these are having a transflorates. It contains not only disc floret and array florets, but it also contains a row of transfloret, hence it is called as a Transvaal daisy. And Barberton daisy, this is mainly because of the place, and also African daisy, it's based upon the place from where it has been originated, hence the origin of this Gerbera is South Africa. And coming to the plant morphology, as you all seen, I think seen the Gerbera plants. So Gerbera plants is generally a tender in nature, and it is a stemless, so you can't find any stems in that. And the plant is completely a perennial in nature, and it gives a flower throughout the years. And this Gerbera belongs to a family Asteraceae, and Gerberas was named in honor to the German botanist and also naturalist, that is Trogard Gerbera. So based upon his name, so they have to honor him, so they have mentioned this crop as a Gerbera. And it was first discovered, this plant was first discovered in 1878 near Barberton, hence it is also called as Barberton daisy. And coming to its usage, so as it's already said in the past slide, so Gerberas are used for different purposes, like the mainly the flowers are used as a cut flowers, used for flower arrangements. Then apart from that, the whole plants can be used in a displaying for flower beds, and also border plants can use as a border plant, and individual plants can be planted in a pot, and it can be used as a pot plant also. And this is one of the best plant for rock gardens. So when there is a crevices or the small pockets which is present in between the two rocks, so there we can fill the media, and we can go for display of this Gerberas. And you have seen, I think, Gerbera plants in front of a college garden also, that is in a waterfalls, so they have planted these Gerbera plants, so it is suitable for rock garden also. And flowers are mainly used for flower arrangements and also floral displays, so these flowers are generally used. Then coming to its importance, so it is a 5th important cut flower in the world market, so that is in a world trade. And coming to its availability of colors, so the wide range of colors are available, and if any colors is not available, so we can go for tinting and produce a desired colors in case of Gerberas. Then apart from that, generally these flowers are used for cut flower purposes, and because of it is having a longer vase life, and also it is easy for rehydration. Then in coming to in India, so the species is distributed from Kashmir to Nepal, that is an elevation of 1300 to 3200 meter above mean sea level. So you can find some of the species of this gerberas in India. Then apart from this, commercial cultivation of these gerberas are generally done in USA, Netherlands, England, also Belgium and Germany. Then apart from that, in India, so it is generally cultivated in Pune, Coimbatore, Ranchi, and also in Erkard regions, and also in Bangalore and other parts. So you can find a commercial cultivation of this gerbera, and these are the places some are suitable for cultivation of gerbera in India also. And coming to its a floral morphology and biology. So as you all know that, so generally the flowers of gerberas are distinguished into three morphological parts, that is ray florets, marginal ray florets, central disc floret, and the intermediate, that is in between the disc floret and the ray florets you can find a transflorets. Then the coming to the sex form of these florets, so the mature ray and transflorets. So these ray florets and the transflorets are having only a female sex organs because due to the poor anther development. So in the developmental stages, so the anthers are poorly developed, and only you can find only the female organs are developed in this. So hence these are female flowers. And coming to the disc florets, so it is bisexual in nature, so presence of both fertile stamen and also the stigmas are present. And coming to this ovary structure, so generally the disc is having a inferior ovary, and the florets are generally actinomorphic, so that is radially symmetrical you can find in case of this gerbera disc florets. But when we come to this ray florets, so the ray florets are generally zygomorphic in nature, that is bilateral symmetry. So if you cut the one ray floret, so it is of a equal symmetry in nature, and also it is also having a inferior stamens, that is male part is generally inferiorly developed. And coming to the complete inflorescence, so there is complete inflorescence, you can call it as a head or a capitulum. And generally the individual florets, if we take a individual florets, so generally it has a 6 to 12 petals, and base of the corolla surrounded by the pappus bristles. So these are just like a hairy like structures which are involved are useful in the transfer of seeds for easy dispersal of seed, so these helps. And coming to the whole morphology, sex morphology of these gerbera flowers, so generally the whole inflorescence is considered as protogynous, that is female is the first, since the flower opening, so here there is a n number of flowers. So individual ray floret is a flower, and also individual transfloret is flower is a separate flower because it contains a separate sex organs. And disc floret, so individual disc florets are also having a is considered also as a individual flower. So individual florets will opens in a different times, and generally the opening of flowers is considered as centripetal, that is opening towards the center. Okay, so the first opening starts from the maturation, the first flower maturation starts from outside, that is ray florets starts opening first, the female flowers opens first, and later after opening of this all the ray florets, then transflorets opens, and later it moves towards the center, and the male flowers will start maturing. Then they also studied the histological studies that reported that 85% of this male sterility exists in case of gerbera. So in gerbera you can find 85% of male sterility existence. Then coming to its genetics, so as you all know that the gerbera jamisoniae, it's a having a chromosome number of 2n is equal to 50, and generally the tetraploids which is produced by colchicine treatment. So generally by using a colchicine treatment in case of gerbera we can produce a tetraploid plants, so that is improving the ploidy level, so increasing the ploidy level. So that is it's also called as colchi tetraploid because the colchicine is used for development of tetraploid, hence it is called as colchi tetraploid. And generally as you all know that, so the tetraploid means so chromosome number is increased. If chromosome number increase means so it will helps in improving the plant character, so plant height and other things, and also flower size will also increase in case of tetraploids. And coming to the plants, generally these complete plant gerbera plants can be divided into three different types, three different categories. So the one which produces completely self sterile, so you can't find a self that is seeds in this, and the second one is the plant with a distinct difference in the seed set. So there is a difference in the seed set when the pollination takes place within an inflorescence. If they in the same flower, so if the pollination takes place, so the seed percentage is different. If the pollination takes place from the neighboring flower which is present in the same plant, so then the seed set percentage will be different. So there is a variation. And the third type is the plant in which these differences are less pronounced, that is you can't find these kind of difference in those plants. And generally for hybrid seed production, this third category of plants which is there, so these kind of plants are generally not used. And coming to the quality and also quantitative differences, it's generally based upon the flavonoid chemical that is present in the cultivars, which will decides the color of the flower in case of Gerbera. And apart from that, generally these flowers are can be also be classified as another three types, that is a single and also semi-double and also double. But majorly you can see singles and also doubles in case of Gerbera. So singles means only 1 or 2 rows of ray florets, and a double means more than 2 rows of ray florets, then it is called as doubles. And coming to the pollination behavior, so generally the pollination occurs in case of Gerbera is through butterflies and also moths. Then apart from these, so some of the bees can also be attracted because of a dark colors or the bright color which is present in the Gerbera. So it will attract the bees and other insects for pollination. And coming to its fertility status, so generally the outer petals are generally infertile because it is having only a female organs, and the central flowers are completely fertile because of its hermaphrodite in nature. So seed set takes place both autogamously and also heterogamously, so that is both by self pollination also and also for cross pollination the seed set will occur. But the quality of seeds are generally higher if it is cross pollinated, and if it is less in case of self pollinated crops, so the quality will reduces. Then self pollinations also occurs, results in a few viable seeds and poor plant characteristics. So generally in self pollination also seed sets, but their viability is very less, and also the plant character will be decreased or reduced, so you can't get a superior plants in the F1. Then hand pollination, so you can get a higher seed set from single inflorescence. Then cross pollination, so more akins than the self pollination, that is when compared to the seed number, so in cross pollinated flowers you can get a more number of seeds than compared to the self pollinated. Then coming to the different species of this Gerbera, so there are different species of Gerbera, that is approximately about 45 different species are present in case of Gerbera, which can be used for breeding purposes. And these are some of the prominent species of Gerbera which can be used in case of breeding and other things. So the first one is Gerbera jamisoniae, it's a commercial one. The plant is generally hairy in nature, so leaves are radiated out from a central growing point in a rosette fashion, that is from a central point, so you can find a more number of leaves and also flower coming from a single node. So from the center you can find a different leaves and also flowers coming. Then flower stalks are generally 40 to 45 centimeter in length, and topped with a solitary orange scarlet head, that is so at the top you can find a flowers, and generally the flowers last up to 4 weeks on the plants. Then single double flowered cultivars and also hybrids can be available in case of Gerbera jamisoniae, both single and doubles can be found. Then the second one is that is Gerbera aurantiaca, so it is generally a low growing herb with a thick long roots. Leaves are dark green in color, lanceolate to oblong, acute, then 12 to 15 centimeter length. So the flower heads are pink orange, and anthers are generally yellow in nature. Then flowering occurs generally in September and November. Then the third species is that is Gerbera ambigua, so the flower colors are generally pink to white. Then beautiful, it's used for beautiful for garden displays and also in grasslands, so these can be used. Then flowers are not single but head made up of a tiny florets massed together, so the ray florets are generally the pink, orange or white colors you can see. Then coming to the another species, that is Gerbera viridifoliae, so the leaves are elliptical in nature. Then flower stalks are short, flower heads are dirty white and small in nature. Then the fifth one is that is Gerbera phyllocoeloides. The leaves are ovate and also flowers are pink, white, yellow, and generally it is grown about 500 to 1900 meter above mean sea level, and blooms in spring and also in summer, and it is prefers a cold, semi shade and also moist soil for its growth. Then other species are, that is Gerbera crocea, Gerbera kanziana, Gerbera tomentosa, Gerbera picta, and also Gerbera tuberosa. So these species can also be used in a breeding purposes. Then coming to its genetics, so as you all know that, so the Gerbera is diploid, that is 2n is equal to 50, so n is equal to 25. Then prior, that is he reported the size of a plant and also flower is more in case of tetraploid than the diploid, that is due to the increase in chromosome number, so the plant character will also increases, like plant height will increases and also flower size will increases. And generally these tetraploids are produced in case of Gerbera using a colchicine. So colchicine is the main chemical which is used in chromosome doubling in case of Gerbera. And apart from this colchicine, they have also tried original, and this orizaline is also prominent. It can be also be used in case of chromosomal doubling. But coming to the study, that is for confirmation of chromosome number, so they have studied in 6 different hybrids, that is Cariba, Amazon, Pink elegans, Terrafame, Aztec, and also Tennessee. So these are the 6 hybrids, so they have evaluated for the chromosome number study. And apart from these hybrids, they have also used some of the non-commercial accessions, so some of the varieties which is still under trial. So accessions they have also tried, that is A7, A8, A9, A10, A11, A12 and A13 of Gerbera species, and almost all the accessions showed a 50 chromosome number. So hence they told the diploid chromosome number of Gerbera is 50. And indicate that the morphological variations in the capitulum, that is single type, double type, and also semi-double type. So this is not having any relationship with the chromosome number. So chromosome number is entirely dependent. If the chromosome number increases, then the flower produced are double. So you can't like that predict. So chromosome number and this floral morphology is having no relationship. And also the mutation and also polyploidy and other things, it is also having a no relationship with the floral morphology. Then coming to the inheritance pattern, so that is inheritance of flower characteristics. So the inheritance of flower characteristics are generally governed by a additive genes, that is the single gene, it's controlled by a single gene, and the expression of this gene is not controlled by any other gene or any other alleles. So generally single gene is involved, and the expression of that gene will helps in development of the colors in the flowers. Then coming to the inheritance of flower color, generally it is controlled by 4 different factors, that is if it is having a allele, then it causes a intense red color. So if it is having a small a allele, then it produces a dull red color flowers. So if the allele is capital H and causing a and a, that is red, intense red, and also dull red. So this is if H is present, H allele is present, then it produces still lighter color of red in case of Gerbera. Then if B is present and which becomes evident in the absence of A, that is in the absence of this intense red color producing allele A, then it can produce a ivory or a yellow color in case of Gerbera. Then apart from this, they have also studied the anthocyanin pigment pathway in case of Gerbera, and generally the anthocyanin pigment pathway contains 2 different enzymes, that is one is colchicone synthase, that is CHS, and also dihydroflavanol 4 reductase, that is DFR enzymes. So they studied so which enzymes is controlling the pathway of this anthocyanin and helps in development of different floral organs in case of Gerbera, and they found that this DFR expression profile vary at different levels of inflorescence. So the DFR expression will vary with the growth of the flower. So in the floral organs development, so different expression of this DFR we can see in flower type and also region within the corolla during the inflorescence development. So there is a difference in the expression of this DFR, hence there is a changes in the flowers. So according to this anthocyanin pigment pathway in different cultivars. Then Patil and co-workers in 2002, they have also studied the shelf life. So if shelf life of a individual flower, so on what it is associated. So generally they found that the shelf life of Gerbera cut flower is associated with the thickness of a flower stalk. If the flower stalk thickness is more, then the shelf life of the flower is also more, because it will imbibe, it will take up a more amount of water and it will last for a longer period. Then they also studied the heritability for different flower characteristics, and they studied that heritability was high for flower diameter and also number of ray florets. So that is heritability means, so from parents to offsprings, so these characters will transfer for a more period, that is a more quantity, that is flower diameter and also number of ray florets, and low in case of disc diameter and also stem length. So you can't get a same stem length in the F1 types, so there is a chance factor. Then heritability estimation results suggested that most genetic variance for flower yield is additive, that is it's controlled by additive genes, that is a single gene it is controlled, and generally for flower yield it's more genetic variations, so there is a variations between the parents and also offspring. Then Harding and Corker in 1991 estimated this additive gene and also dominance of maternal paternal compounds of variations for yield and also flower, disc and ray and also transflorets and the leaves of Gerbera hybrida. So they have studied the genetic variations in case of flowers, Gerbera flowers, and in another study a high heritability combined with a high genetic advance was observed for weight of ray florets, disc diameter, fresh flower weight and also stalk length and also the inflorescence. So all these are related, that is related with a high heritability and also combining ability. Then coming to heritability for post harvest longevity. So the development of cultivar with a superior post harvest longevity is an important aspect. So generally, so we are supposed to develop a variety with having a more amount of shelf life or a vase lives. So in this study, a genetic investigation in the vase life of Gerbera, that is Ashwath, observed that a high degree of environmental influence was found to influence most of the characters. So generally this environmental factors is generally involved in the variations of this vase life in case of Gerbera than the genetic one. So then coming to the breeding objectives in case of Gerbera, so generally for any flowers, so we need to have some of the objectives for before going for breeding. So in Gerbera also they have set some of the breeding objectives. So the first and foremost one is that is uniform and improved plant growth. So generally in case of Gerbera, if we are planting a single variety, so all the varieties should, so all the plants of that variety should produce a flowers in a single time, so that uniform flowering should be there, and also the improved in the plant growth. So generally if the plant growth is compact, so compact growth habit, then it will helps in accommodating more number of plants per square meter. Hence the plant should be compact in nature and uniform in flowering. Then apart from that, more number of flowers per plant, so that we can get a more number of flowers per plant and also yield will also increases. Then novel form and also fancy color. So novel form means so single type, double type, semi double type, frillet type of petals. So like that, if we get any novel type, a new kind of petals and also new kind of shape, size and other things, so then it is a useful for the industry. And also fancy colors, so that is the development of new colors which is not in the Gerbera, so we can develop a new colors. And also structural stem length, so the stem should be straight in nature and the flowers should be horizontally placed on the stems, which can be used for a floral displays. And apart from that, no stem bending and remains upright in the vases. So that is during display purposes, stem should be strong enough to take the weight of the head, and it should not bend easily. So if it is standing straight for a longer time means, so it will fetch us a good price and it can be kept for a longer days in a vase. Then longer keeping quality, and that is nothing but a vase life. Then variety for pot culture, so we can breed a variety which is that is compact growth habit, more number of flowers and also less number of leaves, and less suckering habit in case of pot culture, pot plant. So that is suitable for cultivating in a pots. Then compactness and also uniformity in the growth. Then resistant to Phytophthora diseases and also resistance against the insect pests like white flies. So the major pest which is seen in case of Gerbera are generally white flies and also leaf miner, so that can be reduced. So if we produce any resistant variety. Then coming to breeding methods, so in breeding methods, so there are different breeding methods are available in case of applied in case of Gerbera. So the first and foremost one is that is easiest method, that is introduction, selection. Then the third one is hybridization, then mutation breeding and application of biological tools, that is biotechnological tools. So the first and foremost one coming to, that is introduction. So as you all know that, so introduction is the easiest method, that is we are importing a different kinds of cultivars from a different places So from different countries and other things, we are going to introduce a new cultivars, and then we go for evaluation of these cultivars, and later we can release these cultivars as a new variety for suitable for different regions. So these are some of the varieties which are introduced, that is Algro, Arbo, then Blackheart, Blue Bay, Camilla, Congo, Dilemma, Glory, Golden Gate, Golden Spot, then greenish yellow. So these are some of the varieties which are imported from different places or different countries, and generally in case of this Gerbera, the breeding work is done very less. Hence we imported different kinds of varieties from different countries. And if presently the growing cultivars, so the commercial growing varieties are generally imported from different countries, and it was multiplied in a companies like Florence Flora, KF Bioplants. So there they are going for multiplication of these plants, and they are distributing for the farmers. Then apart from that, this coming to its collections, so different varieties and germplasm has been collected, and different kinds of breeding works is going on in different research institute like ICR research complex from that is northeast regions, that is Shillong. So there also some of the work is going on this Gerbera, and this horticulture research station Yerkad from TNAU. So there also they are conducting some of the work in case of this Gerbera. Then apart from that, GB Pant University, then BCKV Mohanpur, that is West Bengal, then Birsa Agriculture University Ranchi, then Punjab Agriculture University Ludhiana, then Mahatma Phule Krishi Vidyapith Pune, then that is Shere Kashmir Institute of Agriculture Station and Technology, that is Srinagar. So there also they are doing some of the work on Gerbera. Then apart from that, in Karnataka also Indian Institute of Horticulture Research, that is IAHR, is also doing good work in Gerbera, and they have released some of the hybrids and also some of the varieties. Then coming to selection, so the introduced varieties from different countries, so among them we are going to select a prominent one and also promising cultivars, and then we go for evaluation and later released into the public domain or for commercial cultivation. And these are some of the varieties which are selected, like Calcutta Pink, Calcutta Red, Calcutta White, Calcutta Yellow, then GS Lal, Kalimpong Pink, Kalimpong Red, Pride of Sikkim, Priyadarshini. So these are some of the varieties which are developed through selection, that is from the germplasm. So we have selected a prominent one. Then this Arka Krishika, it was released from IAHR, so it is suitable for open field cultivation and yellow in color, so the flower color is yellow. Then coming to the hybridization, so the breeding of this Gerbera was first started in 19th century in Cambridge, that is in England. And when the two South African species, that is Gerbera jamisonia and Gerbera viridifolia, was crossed by R.I. Lynch, and he named the hybrid as a Gerbera canterburensis in the initial days, and later at presently we are calling this hybrid as a, that is Gerbera hybrida. And this Lynch released a variety called Brilliant. So this Brilliant, the variety was developed by crossing Sir Michael Foster into Gerbera jamisoniae. And presently the Gerbera cultivars with different shape, size, colors, and also different colors are generally present and also developed through hybridization and also mutation breeding. So then coming to combining ability, so generally De Jong, that is he analyzed the combining ability for post harvest life in case of Gerbera by taking a 12 parents. So by taking 12 parents, he did a diallele crossing, diallele mating. So diallele mating means, so these 12 parents are crossed with all possible combinations, and the F1 which is obtained, so the F1s are he evaluated. So for different characters in these characters, so he gave more prominence for post harvest longevity. So post harvest characters he taken, so that is first one is, that is days to wilting or folding, then percent folding, then stem curvature. So these are the three main characteristics. So he considered for a vase life or the post harvest quality, and the percentage of folding and stem curvature were examined, and he made a conclusion that folding results in a shorter vase life. So if the folding of the folding days is early, then the vase life is also lesser. Then and also the higher curvature may increases the incidence of folding. So if the stem curvature will increases, then the incidence of folding will also increases, and finally it results in a shorter vase life. And he also studied tested 12 parents of 80 progenies for days to bending or wilting of petals of gerbera. Then large difference in the days to wilting was noted, and it was suggested that the choice for a major factor in vase life. So this wilting or a bending, so these character can be considered as a prominent character when we are going for selection of a varieties for a vase life or a post harvest life. So we can take this character as a prominent one. Then apart from that, they also studied the heterosis study. So that is, heterosis means it is nothing but a superiority of the offsprings over the parents, so both the parents. And in this the crossing experiment was taken by Chiva in 1974, and observed that the heritability of lingual length and the stalk length showed the higher level of heterosis. So the transfer showed the F1 was more lingual length than compared to the both the parents, and the lingual length was found to be a little affected by the environmental conditions. So that is generally for any expression of the character, generally both the characters, environmental and genetic, both are dependent. So in this, the lingual length is completely depend upon the genetic nature. So it is having no role of this environmental condition. So environmentally it's not affected, and to be well correlated with a certain other flower character. So it is used in a breeding program for rapid and early choice of the plants for the commercial use, because it is not affected by the lingual length is not affected by the environmental nature, hence it can be considered as a prominent one. Then breeding for flower quality and also post harvest life. So for this post harvest life and other things, so they have taken a total 69 crosses were made of same color in order to obtain a hybrid with a color uniformity. One hybrid, that is K1 bar 1 into Brigitte, was selected, in which all the inflorescences were yellow in nature. And also they studied some of the top cross mating, that is of either of the parents. So they went for mating, and the scheme used to screen the method to determine the longest vase life among the 31 parents. And the cultivar apple blossom was used as a male donor because it's an excellent vase life and low intraplant variations. So between the plants, so there is a less variations in the apple blossom cultivars, and this is having a excellent vase life, hence this is used for breeding of this vase life study. And in this 5x5 diallele cross was performed, that is 5 parents are crossed in all possible combinations, and the resulted progenies performed very well, and also they showed some of the increase in the vase life by 3.4 days then compared to the other hybrids parents. So coming to some of the achievements of hybridization, so different varieties have been released through hybridization. So these are some of the varieties which are released from private company, that is Runa, Party Time, Purple Queen, then Liang Fen, Autumn Day. So these are some of the varieties which are released from private sector, and these Arka Ashwa and Arka Neser, so these are released from IAHR. So these two are released from IAHR. So coming to Arka Ashwa, it is a cross between IAHR 3 into mixed pollen of 10 varieties followed by selection. So after crossing and F1 development, so they went for F2 and they go for selection of the prominent one, and this variety was released during 2016. The flowers are generally double type and red purple group, then performs well in polyhouse condition with a 50% shade net, also it can perform very well. Then coming to the flower diameter, that is 10.85 centimeter, then stalk length is 61 centimeter, and it is suitable for as a cut flower. Then coming to Arka Neser, so this is a cross between IAHR 1 into mixed pollens of 10 varieties followed by selection, and this is also released in same year, that is 2016. So the flowers are double type and red group, performs well in a polyhouse with a 50% shade net. Then flower diameter is 10.43 centimeter, then stalk length is 61.11 centimeter. The cut flowers and also used for floral arrangements. So apart from cut flowers, it can also be used in a floral arrangements, and also for development of Phytophthora resistant varieties. So the developmental work for the resistance has also taken place in the early breeding programs, and hybridization followed by selection resulted in a 12 variety, among which Akhtur, Migra and Athari showed a resistant to Phytophthora. So among these 12 varieties, these 3 varieties have shown resistant to Phytophthora, and then these plants were cloned, that is through in vitro propagated and compared in screening trial, and 6 showed a different resistance level. So in these clone plants, so the 6 plants showed a different kinds of resistance. Then coming to polyploidy, so as you all know that, so polyploids, that is increasing the chromosome number, can be done using a colchicine in case of Gerbera. But apart from this colchicine, they also tried a oryzalin, that is the tetraploids of Gerbera obtained from colchicine seed treatment increase the plant height and also flower size. And generally these tetraploid flowers which we produce are generally fertile in nature. Then Tosco and et al. In 2000, that is 1995, they have treated these haploid Gerbera plants with a colchicine and oryzalin, that is both the chemicals they have used separately, and they for 48 hours, and they found that this oryzalin is also effective as a colchicine. So this oryzalin is also acts same as a colchicine, that is doubling the chromosome number, and this oryzalin is also having a less phytotoxicity than compared to the colchicine. Hence this less phytotoxicity means the less number of plant will die due to the treatment of this oryzalin. Then apart from that, some of the biotechnological work have also done in case of Gerbera, and generally the commercial planting material which we are getting in case of Gerbera is through biotechnology, that is tissue cultured plants only available in the market. Nobody is going to plant a suckers or any seed propagated flowers in Gerbera plants in case of Gerbera. So only the tissue culture plants are commercially used for cultivation. Then coming to this rapid in vitro shoot multiplication, that is for rapid multiplication. So they used a MS media supplemented with a 0.7 milligram of thiodiazouron was used in Gerbera, that is variety pink star. A large number of green leaf and also maximum shoot elongation was observed. Then apart from that, the in vitro propagation of 3 varieties of Gerbera from the flower buds flower stalk was used as an explant was carried out, and the induction, and it was observed that the flower bud stalks were suitable callus induction and regeneration of shoots. Variety with the red petals showed a best response in case of producing a multiple shoots among the 3 varieties. So among these 3 varieties, so the one which is having a red petals, so that showed a best for regeneration by multiple shoots. So if multiple shoots means in the second cycle we can get a more number of plants. Then coming to somoclonal variations, so somoclonal variations, it is nothing but it's a genetic variations observed among the progenies of the plants regenerated from the somatic cells when it is cultured on a in vitro conditions, so under protected condition if it is cultured. So generally these somatic variations arises due to the genetic changes, and the seeds of this European strain Gerbera were germinated in vitro, and shoots are treated with a gamma rays and also colchicine. So generally they used that is 1 to 4 kilorads, and the plant survived but the growth decreased compared to the control. So in case of 1 to 4 degree kilorads, so the plants are good, but their survivability is less is good, but their growth is not proper. Then the plants were treated with 1 kilorads. The survival of a flower was 100%. And when the same plants when the same variety plants are treated with 2, 3, 4 and 5 to 10 kilorads, so the cause is that is only the survival of flowers is 25%, 46%, 50% and also 100% if it is treated with a 5 to 10 kilorads. So then irradiation of 1 kilorad for 1 to 3 times results in the abnormal characteristics like stunted shoots, then division in the leaf at apex, then narrow and thin leaves. Some florets had red strips on yellow ray florets, and also longer flowers than the control can be seen. Then apart from this, somatic mutagenesis is also studied in case of Gerbera, and according to this the somatic mutagenesis is nothing but a change in the DNA sequence of the somatic cells of multicellular organisms, which is dictated by reproductive cells. So generally it occurs in variations in the DNA sequence. So in somatic mutagenesis is a study of shoots. So the shoots is used for in vitro raising of plantlets in case of cultivar Rebecca, and were irradiated with a single dose of 20 gray gamma rays, then micropropagated for 2 cycles. It causes 25% reduction in the propagation rate in the each cycle. So for every cycle, that is for every second generation cycles, so there is a reduction in the 25% plant survival. So in MV2 stage only 3% plants shown a stable mutation, so they have stably maintained the variations, and apart from that, so 97% have shown a chimeras. So they just shown some two different kinds of genetic genotypes, and that can be reverted back. So we can't take these chimeras for releasing as a varieties. So generally only a 3% they have showed a stable mutation that can be taken for a further, and it can be evaluated and released as a new variety in the future. Then apart from that, coming to molecular breeding, so they have studied this micropropagated plants, genotypes, and also true to type plants is also studied by using a markers. That is, they used a RAPD marker, that is a randomly amplified polymorphic DNA markers they have used to know the true to type of the plants which are micropropagated. And coming to the development of this transgenics in case of gerbera, so some of the transgenic varieties have been developed in case of gerbera. So the first transgenic plants in case of gerbera was produced from the cultivars Terra regina, so carrying the antisense chalconase in synthase gene for alteration of flower color. So for changing the flower color, they have introduced this new gene. And in another study, various this Agrobacterium tumefaciens strains, that is LBA 4404, EHA 101, EHA 105, that is different strains of Agrobacterium tumefaciens were used with a binary vector containing GUS and also NPT 2 selective marker gene for transformation of gerbera cultivar Mariola, and the highest this transformation efficiency was achieved using this Agrobacterium tumefaciens strain EHA 101. So these showed a highest transformation than compared to the other strains. Then about 10 stable transgenics were regenerated from 200 explants. So finally they got a 10 stable transgenics, that is having a new kind of gene in that. So thank you. So this is regarding gerbera. So any doubts in gerbera, so please text in the group.